Okay. Well, the fact that the mouse work, I can just point to things with the mouse. So
the pointer
yeah,
can advance your slides too.
yeah. I may just, since I'm going to stay closer to I'm really just going to try and use that mic.
Okay.
And this only if something is.
If you need to come around and like point or something, yeah.
Yeah.
I'm just going to put a brand new battery in it. Do you have any questions before I have to head back? Good luck. Thanks.
Oh, you might you said it. Nice to meet you. Nice to meet you. Where are you located? Okay, great. So I have to teach you something. But I'll catch up with you. I'll make sure that we talk.
I'm in Alabama.
Yeah, I'm school.
I hopefully it won't be long to calm down.
Yeah, that'll be good. So that's just the first touch with the whole, see what I mean? Let's keep up the communication. If you want to come back here, use for us. The construction labs. We use for everybody to collaborate.
All right, cool.
So but I'll catch up with you at some point. And
Other people can call in if they want. I'm told to use a microphone, so the last thing an Australian needs is a microphone. So it's my pleasure today to introduce Dr. Matthew Waters, who is a new faculty member at Auburn University. He's a paleolimnologist, so he works mainly on reconstructing histories in aquatic sediments and such. So very applicable to what we do here at the Sea Lab, so I think a lot of you will find some interesting research ideas and questions that come up with this. He did his bachelor's degree at Mercer University, which I think is in Georgia. Okay, I'm getting better. And then his master's degree at the University of Florida in aquatic sciences. Then he went on to do his PhD in environmental sciences at UNC Chapel Hill, which was in 2007. And he then did a postdoc at USF for a year before becoming a faculty member at Shorter University back in Georgia, and then at Valdosta State, which he's now back at Auburn, which is fantastic. I always like to include some more personal facts, though, when I introduce a speaker, because I think that shows us that he's also human and not just this very intelligent, fancy paleolimnologist. So he has had a pint of Guinness in 19 pubs in Dublin. So that's quite a feat in its own. I'm not sure if that happened in one night. He's a small guy, so I hate to think 2 nights.
I think, if I can remember.
Nice. He likes to brew beer, and enjoys craft beer. And he also plays the acoustic guitar, banjo, and mandolin. So he's a man of many talents. And he's also extremely busy. On top of all of this, like doing research and playing instruments and brewing beer, he has a wife and 3 kids. So that's going to keep a man busy no matter what. So anyway, with that, I would like to welcome you, and we appreciate you giving us a seminar today.
Sure. Thank you. So I'm told I have to use a mic that's loud. Is that too loud? Are we good? I can be loud. Okay. All right. Hey, everybody. How we doing? If you can see me, I'll be on my tiptoes maybe. More on that in a minute. So I have an agenda. I am a freshwater scientist primarily, but I do a lot of these connections, and where I work in these freshwater environments, I look at the complexities of these freshwater environments, and all of a sudden I look up and they're going toward the coast. So my objective today is really to just sort of describe some of these systems that I've been working in, let you know some of the complexities that these systems hold, and just make you aware that these materials are coming toward the coast. We got to work together. We got to figure out how to solve these problems, or we got to look at how these impacts are going to occur and study them together. So that's sort of my objective. I'm not a marine scientist. I like to say I sort of play one on television because I hang out with a lot of marine scientists because y'all are cool. If you look at my collaborators up here, a lot of these people are Marines. Some of y'all might know Mike Peeler or Chris Martins from UNC Chapel Hill, Institute of Marine Sciences, USF St. Petersburg. I work with Donnie Smoke. He does a lot of my lead 210 modeling. And then a whole host of people. So I'm sort of presenting to you 3 different projects this morning. So I do want to thank my collaborators and my graduate students that have worked on this. So we got to connect us. We got to connect inland waters and the coast. And that's nothing new. Any hydrologic cycle in any textbook that you open up is going to say that we're all connected. The materials are moving from the landscapes into the freshwater systems, from the freshwater systems into the coast. This is obviously well known by people who've been studying the Mississippi Dead Zone for a good bit of time. But down here in the bottom right, John Cole, who's a famous biogeochemist of carbon in the freshwater world, really started looking at the way that the movement of carbon in freshwater systems is much more complex. And this is just a quick figure from his 2007 paper on the subject. And it's not just the landscape is moving things into freshwaters, and freshwaters are just moving things to the coast. There's a lot of processes involved, processes of deposition, processes of respiration and the movement of gases into the atmosphere. And this is really sort of where I want us to think about, not only for carbon's sake, but for phosphorus and nitrogen, and also some of the nature of these carbon species, meaning whether or not we have algal material, cyanobacterial material, or macrophyte material. And that's what's moving towards the coast. And the more that I have uncovered a lot of these freshwater systems, the more I've started to realize that the stories are unbelievably complex, the management is going to be complex, and that's something I hopefully can unpack today. This has been made highly evident in recent news from the St. Lucie River in Florida. They have had a cyanobacterial bloom there that essentially smells like sewage, from what I understand. It's gotten very nasty. If you listen, my old NPR station was from Florida, so I heard this all the time. People from this area of Florida, this is on the East Coast, have been taking bottles of this water and putting it on the governor's desk because the governor of Florida will not come down here. And this is a coastal problem. They're showing pictures of fishermen not being able to use their boats. But really, this is a freshwater problem. This is a freshwater lake problem. And the cause of this algal bloom is Lake Okeechobee, which is in the center part of the state. And what happens with Lake Okeechobee is it's in this agricultural watershed, and it's receiving a large amount of nutrients over decades. And the government of Florida is trying to buy up this area, this agricultural sugarcane area now, and shut it down and remove these nutrients. But the problem is, and this is going to be a take-home message of the day, is Lake Okeechobee is a shallow lake system. And shallow lake systems work completely differently than most other lake systems, in the fact that these sediments that are decades old in the sediment record can be resuspended and can move. And that's what's happened. This legacy phosphorus, this legacy nitrogen has been resuspended because what happened in this situation is the El Nino waters from all the precipitation filled up Okeechobee. They had to move the water out to the coast to get rid of it, and they did, and it caused the algal bloom. And that's sort of the take-home message of what we're talking about. We're talking about shallow ecosystems and the way to manage them. So just a brief lesson in shallow lake limnology. When we look at a lot of things in the limnological world, this is what you get in a textbook. You get your standard lake made by a glacier, studying in Wisconsin or in southern Canada, and this is what every textbook is going to tell you. Materials are coming in, we monitor total maximum daily loads, we figure that out, and then it's going to be deposited down in the sediments. That's not what we have in the southeastern United States. Most of our systems are shallow, and what you get is you get the ability to resuspend. You get the ability for whatever gets into these systems is going to be churned up again. It's going to be mixed during wind events, during mixing events. You know, just by looking at the bay, I'm sure you all know about mixing events and wind events and the movement of sediments. So this is what we have in these shallow lakes. So we're not only talking about just shutting down inputs of phosphorus and inputs of nitrogen. We're talking about legacy nutrients and the impacts that they can have. The other thing is, since we have this rechurning of materials, shallow lakes have the ability to be highly productive, be macrophytes or algae. And this can change at a dramatic rate. It's called the Alternative Stable States Theory of Shallow Lakes. It's a pretty famous paper by Martin Sheffer in 1998. And basically the idea is this, is in a macrophyte-dominated system, the macrophytes, you have clear water, you have increased biodiversity of fish, but also these macrophytes have the ability to anchor sediments. And since they anchor the sediments, they prevent resuspension, you have more resilience in the system to change. But over time, algal communities start to outcompete the macrophytes for light and for nutrients. You start to get a dying off of the macrophytes, and you can have a dramatic shift from plants to algae, usually sometimes within a month, a complete ecosystem reversal. And when you have these algal-dominated systems or cyanobacterial-dominated systems, you get flocculent sediments, you get a decrease in biodiversity, and you get these resuspension events that are much more common. And this is sort of what we're seeing in the southeast with most of our systems, is we're moving towards, it's a story that everybody knows, we're moving towards eutrophication, we're moving towards cyanobacterial dominance, and what happens when this material reaches the coast? That's where I want us to think about. So I work in the southeastern United States in the lake systems there, and usually when I go to meetings and I tell people I do that, they go, "Oh, you work in Florida." And I go, "No, I do work in Florida, but I also do work in other places." And this is, I just came up with this slide just preparing for this talk. Basically, the southeastern United States really is a shallow lake district, is what it is. We don't have deep lakes in this area of the country. And in case you didn't know, there's a large number of shallow lakes in North Carolina that virtually are unstudied. You're familiar with Florida, the panhandle of Florida. If you type in Georgia natural lakes, you'll get nothing, but that's a lie. There's 20 to 30 natural lakes in South Georgia in an area called Lake Park. I've been studying them for quite some time. And if you notice, I include Alabama over here, just out of personal interest, because y'all have Lake Shelby and the lakes over there near Gulf Shores. And you also have a lake in your bird sanctuary. Did y'all know that? Go see it. It's fantastic. I jogged by it this morning, even took some pictures. So we are a lake district. We are a shallow lake district, and the other thing I want to just put in your brain right now is these lakes are near the coast, no matter where you go. It is a coastal system, is what it is. So we have to work together. You got to know what's going on in my world, And I need to work with you to understand what's going on in your world, because we're definitely connected, and we don't want what's happening in St. Lucie to happen anywhere else. And that's where we need to be. So I picked 3 of these systems that I study, and I'm just going to kind of take you through them and put it into your brain so that we can maybe have conversations, or you can have conversations about what's coming down, what's coming toward you, and how we can manage things correctly for the future. And understand these systems. We're going to talk about the Florida Everglades first, very briefly, and then I want to introduce you to Lake Mattamuskeet, which is one of the most interesting systems on Earth, in my opinion. And then I'm going to end with something a little more close to home, Lake Seminole, Georgia, which is a reservoir that feeds into the Gulf, which is much more common to most of your work. So we're going to end there. We're going to talk about some of the complexities that we find there. So that's our stories. So the way I do this is I'm a paleolimnologist. I've studied the history of freshwater inland systems. And the way that we do this is materials move into these systems, usually from terrestrial landscapes, and then we get some autochthonous production inside. These things that move, they die, they sink, and they form the sediment record. We core those sediments. We bring these sediment cores up to the surface, and we section them going back in time. So I have these little hamburger slices, and I can measure a whole suite of chemistries and other things moving down, date those sediments, and I can reconstruct the histories. And we do this through a number of proxies that we measure: organic matter, stable isotopes, total carbon to total nitrogen ratios. Many of this stuff probably is very familiar to you. Nutrients, metals. We date using lead-210 for the past 100 to 150 years, and below that we use carbon-14. And I'm always interested in the response of the ecosystem. We characterize the materials coming in, but I'm interested in the response of the ecosystem to its primary producers. And I do that by measuring photosynthetic pigments using an HPLC system, chlorophylls and carotenoids. And since I talk about that a good bit, and that's a good bit of the collaborative work I do in marine systems, because these work well monitoring algal and cyanobacterial communities. Just to give you a pigment primer, many pigments that I measure are ubiquitous among primary producers. Chlorophyll A, phaeophytin A, beta-carotene, you've probably heard of those. But many of these other pigments are quite diagnostic. They're for specific algal groups. Diatoms, I can look at a couple of pigments called diatoxanthin and fucoxanthin. There's no test involved in this, by the way. Cryptophytes, which are a modal, swimming, small algal organism that usually can dwell with cyanobacteria because they can get up into the light. Is a pigment called alloxanthin. Green algae and macrophytes usually share pigments, lutein, chlorophyll B, phaeophytin B. I even have one pigment, pyrophaeophorbide, which is what happens to chlorophyll A when it moves through the gut of a zooplankton. And then there's one for red tides. K. brevis has gyroxanthin diester. And then I do a lot of work in cyanobacteria, and we measure a whole host of those. A couple that I will point out is a phanazophyll, which is common in infixing cyanobacteria. And then this really interesting called cytonemin, which is a UV blocking pigment. It's sort of like a benthic cyanobacteria sunscreen. So we can tell whether or not cyanobacteria are living on the surface of sediments and whether or not light is intense and is reaching them. So I put all these things together and I can reconstruct algal histories. And that's sort of a lot of the work that I do in my lab. So let's start with the Florida Everglades. Beautiful system, lots of alligators, and now pythons. Unbelievably altered system by humans, and this is going to be, it's not only nutrients that's affecting fresh waters, it's nutrients and hydrology that's really affecting fresh waters, in my opinion. And here's Lake Okeechobee, again, the largest lake in the southeast. And below it is the Everglades agricultural area. And then to move water to municipalities, to maintain water levels, they also have built these WCAs, water conservation areas. So what you have is you have your source of phosphorus coming from the agricultural areas. You have the movement of water going towards municipalities. Remember, this is Miami and West Palm Beach and those types of areas. So you have a management for humans and a management for ag coming down. So it's a shallow ecosystem. It's a large wetland. It's called the Sea of Grass. If you've never been there, please go. Currently, it has constant water levels, and it has been invaded by typho or cattail. And the cattail community is sort of slowly moving southward in these systems. And here's a satellite image, and I don't know if y'all can see with the contrast, but can y'all see there's a contrast change right there on that line? And below here, it's sort of this bluish green on my screen. So this is the cattails. This is the typha moving southward. And this area are what we call marl prairies. So due to that we're in this karst limestone environment, some of it has gotten up, and these algae and cyanobacteria, they will form these algal mats. And I don't know if you can see these little things floating in this picture right here, but that is cakes, literally white cakes of cyanobacteria and some green algae, but it's primarily cyanobacteria. So we have this system that is moving slowly. This is not an acute system. This is a chronic system. It is moving slowly. It's moving southward. And we were hired, South Florida Water Management District. We took a transect of sediment cores through this system. If you can see, we went all the way from typha, that's been there a while, to typha that has just recently invaded, down to the marl prairies. So I'm going to show you the data from that project right now. By the way, if you don't know, just Google pythons in the Everglades. If you're like, why does he have a freaking snake up there? That's why. Just give it a Google. You'll be happy you did. Okay. All right. So if you're not familiar with sediment studies, so here's how it works. We basically present to you the core as we took it. So the top of my figures are all modern day. That's the top of the sediments that we core, and then you're going down core, and on your, what most people call the y-axis, I call it the x-axis. That's our dates. These are lead-210 ages from our dating model, and we use that to compare the three cores. And all of these cores go from the typha, the most northern core, to the marl prairie. And I've never seen more beautiful stratigraphy than I did in these cores. They look like cakes, is what they looked like, because the marl is white, and it has a very low organic matter content compared to the other peat of the Everglades. So you can easily see, especially in the one that's been typha for a while, you have historic sawgrass down here. Then you have a decrease in organic matter. This is, or total organic carbon is what this is. You have a decrease in organic matter. That's your marl with cyanobacteria. And then you have the influx of the typha. The middle core, you have the sawgrass. You have sort of a mushed marl signal here, but then you see it comes back up, and you have the recent influx of typha. And this one you have the decrease, and we do not have the typha backlash yet. This should match up well with cyanobacterial pigments, and it does. This is a figure of canthaxanthin, which is specific to colonial cyanobacteria. And you can see wherever we have marl, we have increases in cyanobacteria deposition into the sediment. So we feel like we're getting a good story here of when we go from sawgrass, which is the historic natural condition of the Everglades, to marl prairie to typha. It's a little bit busier when you get to chlorophyll B, because green algae and macrophytes both use this. So, we have a decrease in chlorophyll B here, an increase with the typha, but we have it in the marl here because, again, some of the marl contains some green algae. So that's not too surprising, but at the same time, we do feel matching all of these stratigraphies together, we have a good definition of the rate. How fast is the typha moving, and at what rate it is moving, we can build off of this. And that's part of the paper that we have in J. Paleo. But I'm interested also in mechanism. What is the cause? What is the speed? What can we measure to predict? We need to be able to predict. And digging through the literature, phosphorus became clearly linked to typha. Sedimentary phosphorus became linked as well. And when I started trying to find some papers, I found one by Wu et al. That gave a number that said 0.65, these are from mesocosm studies, 0.65 milligrams per gram is when typha has the ability to overtake native vegetation and to invade, basically. And then I took that and put that back on our cores, and it matched up almost perfectly. And very rarely, if you're in graduate school right now, very rarely does the data come out like this, okay? So don't expect it. But it did in this case. And it was unbelievable. If you see this line, which is 0.65, wherever it passes over sedimentary phosphorus is about when we switch from marl to typha, 1950, and then right in the top of this core, and then it has not reached here. So we are presenting this saying it's not water column phosphorus. That's what everybody measures. It's not water column phosphorus that's driving this invasion. It's sedimentary phosphorus. You need to be monitoring sediments and seeing how sedimentary phosphorus is increasing. Unfortunately, that was deaf ears that we sent that to. But The story is real, and it's a chronic story. This is not an acute story. Acute story is what gets in the news. Chronic is what sneaks up on you. Sedimentary phosphorus is slowly moving through the system toward the coast. What about the sediment phosphorus reaching some of these canals? Could they move towards the coast at a quicker rate? We need to be looking at that. Current ag is decreasing, but these legacy nutrients aren't going anywhere. And sediment materials are not part of the current monitoring program at all. Still to this day, they're not monitoring sedimentary phosphorus in this area of the Everglades. So it's moving. It's moving slowly. Who knows what is going to happen once it gets closer to the coast, and we're not far. And just in case you're interested, what about the other side of the Everglades? We're also doing some work over there. I don't have any data to present from this because we're just measuring stuff, but we're looking at the opposite way because the Gulf Coast of South Florida is where the Everglades used to export and move its water out into the Gulf. They've changed that now for it all to go to Miami and Fort Lauderdale. So what about the other side? And the other side is a sea level rise story. And what we're looking there is we're taking these pigments and we're doing diagnostic pigments on sawgrass and mangroves, and we're trying to match the speed of mangrove encroachment into the coast. I don't have any data on that right now, but that's another project we're measuring in this area. All right, Lake Matamesquite. Isn't that just beautiful? I took that picture. I just want to take credit. Isn't that beautiful? Isn't that fantastic? Unstudied lake. I mean, this is anyway. It's the largest lake in North Carolina. It's so unique. It's 160 square kilometers, 16 miles by 8 miles. It's a meter deep. It's 1.2 meters deep max. You can walk across this lake for 16 miles. Has a very unique hydrology. I don't know if you can see, there's a couple of lines here that you can see pretty stark. They have canals because right there in the bottom of this picture, that's the Pamlico Sound. This is right by the coast. We are right by the coastal waters here. So basically these canals were cut and they put gates in this lake. 3 of these canals are on the east side of this road. 1 of these canals is on the west. And they put these gates where they let the fresh water out during periods of high rainfall, but they prevent saltwater intrusion to maintain the ecosystem. So it's a one-way gate flow to the coast. Keep that in mind. There's also a little bit of exchange across this roadway. There's some small culverts. There's not a lot of exchange, but there's a little bit. Human impacts. We could write a textbook off of the human impacts of this system. If you look here, what does that look like to you? Lighthouse, right? It's not. We're not there. That was in 1915. That was the world's largest water pumping station. So a Dutch company tried to buy the whole lake, pump it dry, build a town on the lake bed, and farm it. But the problem is the lake bed is below sea level because we're so close to the coast. So 3 different companies went bankrupt trying to pump this lake dry and never succeeded. And then they finally said, screw it, basically. And the government bought it in 1937 and turned it into a national wildlife refuge. If you can see there, it's covered, it's surrounded by ag fields. A lot of those are grandfathered in since the 1800s where there's no regulation. Whatever you want to dump in there, you can. And it's maintained today for one thing and one thing only, and that's waterfowl. So, highly interesting system. We started off here with just a transect through the system, and lo and behold, this idea of shallow lake ecology really popped out, and what we found out is we have completely different ecosystem states on the different sides of this road. The east side is covered in Vallisneria americana, and it's clear water, and it's where the waterfowl like to hang out, and it's a beautiful macrophyte-dominated system. The west side is a cyanobacterial cesspool, Phanozomenon, and a few other critters over there. So we have dual ecosystem states in the same geology, in the same precipitation, in the same land use, in the same management. So obviously we wanted to take some cores and try and rebuild that history. So here's the east core, and I put these pictures. This is the macrophyte-dominated side. This is the algal-dominated side. And the only thing I want you to get from this picture to take forward is, can you at least tell that organic matter, total phosphorus, total sulfur, is depositing and storing a lot more in the west than in the east. You can tell that by comparing these two. They were both on the same scales. We get more materials depositing in the west, the algal-dominated side. Okay? looking at the pigments, which again I was trying to reconstruct the histories. We took all of our pigment data, put them into a cluster analysis, because going back in time, this lake was sand bottom and clear. So this is a unique, we think it's a unique environment. More on that in a second. But so we put these into a cluster analysis to break it up into algal sediments, macrophyte sediments, and low productivity sediments. And then did a principal component analysis with all the pigments. And what we see, it just made sense. We see a good separation of the data. And we see these two pigments, aphanizophil, which is a cyanobacterial nitrogen-fixing pigment, and allozanthin, which is for cryptophytes, which typically dwells in eutrophic environments. And these ordinated with the phytoplankton sediments. And then the macrophyte sediments down here, we had chlorophyll A and chlorophyll B, which are in very high concentrations in macrophytes. So we got a really good separation of our data, is really what I want you to get here. Okay, we got a really good separation of the data. Putting that back onto the cores, let's tell the story. So these low productivity sediments clustered together in the bottom of both cores. So that was really nice. So prior to 1850, basically this lake was a ligotrophic, clear water, sand bottom. You would want to own a house on it. And then it changed, but it changed for both sides. It changed to an algal dominated system. And 1850 is when they cut the first canal to the coast, basically. It dropped the water levels, made higher exchange between the two systems, and as a result, rapidly, the system went to algal dominance. But then it switched only on the east side to macrophyte dominance around 1940, 1942, and that's when they built the roadway. So something happened with the hydrology, something happened with the nutrient movement, the biogeochemistry of the system with the building of the roadway. And if you get into theory, everything is backwards, because the larger lakes should hold phytoplankton because you get more resuspension, you have more fetch. That's not what we have here. It's backwards. Pretty much everything that we could apply that was in the literature was backwards, and we should have plants on the west and algae and cyanobacteria on the east. So really trying to get at the mechanisms, and since this paper, we've shown this through monitoring. But basically, this is the mechanism that is happening. It's not always nutrients, it's nutrients plus hydrology. So the primary wind direction is out of the southwest for this system. And if you remember the culverts, or the canals that leave the system, there's 3 on the east and 1 on the west. So what is happening is nutrients are being flushed out of these canals, which lead to the coast, are flushed out of these canals during periods of high flow and of wind, and you get a removal of nutrients on the east. But then on the west, there's very little connectivity between the two sides. These are only about 10 feet wide, over 8 miles. There's only 5 of them. And what you get is you get a cycling of nutrients in the west, which is why we see high deposition of materials in the west, low deposition of materials in the east, a flushing of nutrients. We get a maintaining of the macrophytes. But if you notice, if you think through this, they are connected. And if nothing is done to manage the system, it's going to change. The phosphorus and the nitrogen is going to get to the east. And that's exactly what we're seeing. This year is the lowest macrophyte count that we've seen since the lake has come under our monitoring. So it's not a good story. Slowly the cyanobacteria are moving east, and it's just a matter of time, unfortunately, just because there's no management in the watershed. So I also want to tell you that we do put these things in historical perspective. So we did go back to Lake Mattamuskeet because we're very interested as to whether or not what these patterns that we are seeing are unique. Are they novel ecosystems? Are they reoccurrences that did occur due to climate changes in the past or other things? So this is a vibrocore system that we use to go get much more mud from Lake Mattamuskeet. This is a 400 centimeter core. And as you can tell, it's very easy to tell the change from lake to marine. And we carbon-14 dated this. It's carbon-14 dead. So we can easily feel like we have the entire lake history captured in this core. And as you can see, just the take-home message here is what is happening since 1850 is very unique. There's no other time, really, that we have this much organic matter, phosphorus, sulfur, and then I've blown up the top 150 centimeters. It's the only time we see really algal communities coming into the system. So what we're finding in these systems is highly unique. They're novel. The environment has not seen these systems before. And they're moving a lot of materials from terrestrial environments into freshwater and potentially freshwater into saltwater. So putting this all together, I just wanted to blow up, just to give you the perspective. Here's Lake Mattamuskeet. If you've ever been to the Outer Banks, this is the Outer Banks, and this is Manio or Roanoke Island, which is where the Lost Colony was, if you're familiar with your American history. Lake Mattamuskeet is actually on the painting that they did for the Lost Colony and took back to England before it became lost. So there's a piece of history for you. But just to give you a perspective of this, here, just following this canal, it goes unimpeded, by the way, through the landscape, which is also agriculture, and hits the coast right here. This is the Pamlico Sound. And then here's this canal where the pumping station was located, and it's a straight shot straight to the coast. So we have connections here. And this plume of phosphorus cyanobacteria is moving from the west to the east and is going to be flushed out. So the implications here are these materials are reaching the coast already during rain events and wind events. Cyanobacteria dominance is invading the east side. No regulations are occurring in the system. And another thing I want to just show you some of the work that my lab is doing is I am interested in cyanobacterial dominance, and if you're familiar with the work that Allison's been doing, these cyanobacteria can produce a lot of toxins, and I automatically think, let's go back in time. So I have done some toxin work in the sediments. There's very little work that's been done in this, but this is a figure from Lake Griffin, Florida, of Cylindrospermopsin, which is a hepatotoxin that some cyanobacteria can produce, and there is Cylindrospermopsin that is 4,700 years old. So some of these toxins have longevity, and they're not just going to go away. This idea of legacy from freshwater systems reaching the coast can occur on the toxin level as well. So a lot of interesting things that can come from this, and we're going to try and start studying some of these outflow areas in the near future. So let's talk about Lake Seminole, which is probably the most applicable to the materials that you guys would receive from inland Alabama. That's a lake, I promise. It's a reservoir, and it's covered in hydrilla, which is an invasive species. And what you need to know is that's about 2 meters deep right there, 2.1 meters deep, and it works through the entire water column. That is not a superficial plant. It is that much biomass all the way down. What ecological role is it having? How is it changing the materials leaving Lake Seminole, moving toward the coast? That's something to keep in perspective. But this is what you'll see if you Google Lake Seminole. They'll say hydrilla, hydrilla, hydrilla. Homeowners hate it. Fishermen love it. Before I get into that, I avoided reservoirs for the first part of my career because I thought they were boring. And then I read this paper by Savitsky in Science, and basically he just opened my mind that we are trapping 26% of the global sediment behind reservoirs right now. And if you don't know you're inland Alabama, we are a reservoir heavy state. We are a ladder of reservoirs in this state. And we are trapping, who knows how much sediment we are trapping that is trying to reach its way down here that is not making it. It's being stopped by these reservoirs, and there's not a lot of research characterizing those sediments. We know total sediment. We know usually maybe some heavy metals in those sediments, but we don't really spend a lot of time characterizing those sediments. And that's of high interest to me, which is really what led me to start working on Lake Seminole. Really interesting story here. Lake Seminole is the confluence of 3 different river systems. The Chattahoochee River, which runs through urban areas of Atlanta and Columbus and is highly regulated. There's 13 dams along this river, and it is a very fragmented system, a fragmented urban system. You contrast that with the Flint River, which is an agricultural watershed. This is the agricultural hotbed of Georgia, if you do not know that. A lot of pivot irrigation, row crops for as long as you can see, and it is almost an unregulated river. There's only 2 reservoirs on this system, but they are both run-of-the-river reservoirs, which means they are not used for storage. They're just used for small hydroelectric plants. So really different systems, and they come together in Lake Seminole, and here's where you come in. They form the Apalachicola River, which runs to Apalachicola Bay, which, as you know, is a huge oyster fishery area that is really, really struggling. But if you look into the news, all they will talk about is fresh water. They'll talk about flows. They'll talk about the amount of water coming into Apalachicola Bay is low, and you have a change in the salinity, which is putting a huge stress on the oysters. And the materials moving down this system, or the carbon moving down this system, or dissolved materials moving down this system is never even in the equation. And I don't have, we're just moving down into the Apalachicola, but once I show you what's going on in Lake Seminole, you tell me if you think it's actually releasing some materials down there. That's where I want you to be. So Lake Seminole was dammed in 1957. It's a pretty large reservoir, and it's a shallow system. All these run-of-the-river reservoirs generally have a lot of shallow areas in it. The management is really threefold. Hydrilla, they try to kill it. Enough to make everybody happy. I mean, to get through this stuff, they literally cut roadways through the hydrilla is how you get through this lake. Dam release, just due to the Army Corps having to release so much water to get to Apalachicola by law, and then the bass fishery is huge. So we collected a core from each side, and I'll sort of build this in the way that we discovered it, because it was a really strange study, and it's taken so many turns, and we're still not done yet. So I took a core from each side, in hydrilla beds, because they wanted me to reconstruct the hydrilla history. When did it arrive? How has it changed the ecology? That was the project that I was brought on. So I took a core from the Flint side, that's the agricultural side, and the Chattahoochee side, and that's the urban side. So the first thing that popped out in this was sediment delivery. Again, you have an unregulated river in the Flint and a regulated river in the Chattahoochee. So if you don't know anything about lead-210 dating, it has a half-life of 22 years, roughly. And when you take a lead-210 core, you really want to see something like this. You see high amounts, which are recent sediments, and then you see some sort of degradation or radio decay based upon time and changes in sedimentation rate. And then you use this to build a model of sedimentation and of dates. And that's how basic lead-210 works. So this is a decent looking lead-210 profile if you're familiar with the systems and you're familiar with the mechanism. And then my friend Donnie Smoke, who does my lead-210 work, he called me and he goes, "Your Chattahoochee core is terrible. We can't use this. This is the worst looking core I've ever seen in my entire life." Because what you have is, you have modern sediments on top of old sediments. If it's excess lead-210 dead, then that means it's over 100, 150 years. You've decayed all of the lead-210. And I said, wait a minute. That could be exactly what we expect, is you have new sediments when there's low release periods. And then when there's high release periods coming from these dam, you're going to have old sediments from construction, from urban areas, from these silts coming down. It's not as organic. So you have young, old, young, old, young, old, young, old. And it makes sense. And we just got this published, and we're doing more, and we're finding this in regulated systems. So what that tells us is sediment delivery is completely different based upon the dam release strategies, is what it is. So yes, we are storing tons of materials behind these dams, but the way those materials are getting to the coast are completely different based upon the dam management. That sounds terrible, doesn't it? But it's based upon the management of the system, and that's not even looked at. That's not even on radar screen. And you're going to be receiving completely different situations based upon the dam release schedules. So that's one thing that popped out. So here I am trying to reconstruct the hydrilla, and I really just thought this was going to be a very boring study. Hydrilla straight down, hydrilla signal. And so this is the Chattahoochee core, this is, again, the urban core, and this is the ag core on the bottom. And again, this is what you expect, high organic matter coming from the ag watershed, low organic coming from the urban, totally makes sense. Then we got to the TOC to TN ratios, which is a measurement of structural carbon. Terrestrial trees have very high TOC to TN ratios, macrophytes have a less, and then algae and cyanobacteria have even less. So in the Chattahoochee core, we saw what we thought we would see. We saw TC to TN ratios around 12.2 for the whole core. 12.4 is what hydrilla has if you just take hydrilla out of the lake and measure it. Completely understandable. Most of the organic matter coming into the Chattahoochee is from hydrilla and hydrilla derived. But when I looked at the Flint side, completely different. It was an algal signal, 8.3. That is very low structural carbon. This lake is clear, y'all. You can see to the bottom of this lake. It does not have algal blooms. Algae is not a problem. You have decent amounts of nitrogen in the water, but it's very, very phosphorus low. Very phosphorus low in the water column. So this was very strange to us. We didn't expect to see this signal. So then I looked at a couple of the other pigments, canthaxanthin, which is a cyanobacteria pigment we've already looked at, and cytonemin, which was that UV-blocking benthic cyanobacteria. And I started going out and taking ponar dredges and grabbing claws of sediment off the bottom. And wherever there was not hydrilla, by the way, we don't see this in the Chattahoochee side, but wherever there was not hydrilla, we found the benthic cyanobacteria lingbia, which is also a toxin producer, which at certain periods in the summer can over photosynthesize or die and rise up to the top, form bubbles, and get out of there. But it's everywhere, and they didn't think it was until we did this research. So we have a benthic cyanobacteria and a macrophyte, but we do not have a lot of nutrients except for nitrogen, very low phosphorus, very phosphorus-starved system. But we have more primary productivity than I've ever seen in any freshwater system I've ever worked in. And I kept telling them, the nutrients are there. They got to be there, but they're just not in the water column. Keep that in your mind. So we did phosphorus profiles, and the results were pretty staggering. So it's not too much in the Chattahoochee side, but in the agricultural side, it comes out to be 2.2 milligrams per gram in the surface, 1.9 milligrams per gram average for the entire lake history of the core. This is phosphorus deposition in the Flint side. Put that into perspective from things that I've already shown you. You see a list of the catastrophic eutrophication stories of the world today. Lake Griffin, Lake Apopka are both famous for pea soup eutrophication. You have more phosphorus in the sediments of Lake Seminole than you do in those. There's Lake Okeechobee that just caused the eutrophication, almost double the amount of phosphorus. And by the way, just to put this into perspective, these are surface samples, and this is an average for the entire core. Okay. There's the Everglades Water Conservation Area 2A. Lake Taihu, China, if you're familiar with that, is a cesspool in China right now that they are really having a tough time dealing with. And just to put another reservoir in perspective, it's almost a fourth. So we have incredible phosphorus in the sediments, but this phosphorus is being sequestered by the cyanobacteria and the macrophytes, but it's also being anchored by all of this material. All of these macrophytes and these cyanobacteria are anchoring these sediments, preventing any sort of release downstream. And again, this goes straight to the coast unimpeded. However, we started measuring hydrilla. We started measuring macrophytes. We're like, we need to get a handle on the dynamic nature of these macrophytes moving forward. And here's a map. Again, we're 160 square kilometers, so this is a lot of biomass. I mean, a lot of biomass. And you can see it's pretty much everywhere. There's not the river channel. You see these pockets of hydrilla, and all of this is hydrilla mixed, basically. And what I want to show you is this is 2012, and keep your eye on this huge, many, many miles sea of hydrilla. And again, this is the biomass we're talking about. Imagine how much leaves, and here we go. Here's 2013, and it all vanishes. The amount of carbon and nutrient plume that must have been released in that, nobody has measured this coming into the Apalachicola Bay, as far as I know, is looking for these types of materials. And then in 2014, we see it coming back a little bit, but it's been very difficult to establish. And what is this doing about the sediment releases? We're seeing some struggles now, on the Flint side, which is where all the phosphorus is stored. Where are these materials going? Is this moving from structural carbon to DOC? What is the carbon dynamics? None of this is being studied. The coast doesn't even know this is happening. I called Sea Grant and said, hey, how about some of this? And they were like, I'm sorry, this isn't sea work. This isn't coastal work. And I was like, trust me, it is. We need to connect this. So it needs to be studied. Who knows where these materials are going, but I hope you can at least see the amount of material is unreal that's leaving in this system. And it's getting through. It's moving through the dam. So what's causing this? What's the driver? Is this nutrients and shallow lake ecology, alternative ecosystem states? It's actually not. This is completely driven by flows. Completely driven by flows. 2012 was a drought year. That's when we had the maximum amount of hydrilla coverage. You can see that in this hydrograph right here. Very low flows that year. In fact, you can see this is almost a straight line because that is the lowest flow that the Army Corps has to release legally. And then you see 2013 and 2014 are wet years, and that's when the hydrilla started to decrease. So we are related to precipitation and dam management in the system. And what I want you to see is flow is driving hydrilla coverage and macrophyte coverage, and macrophyte coverage is driving a lot of the biogeochemistry in the system. And all of this is very, very complex. So the other thing that we cannot forget is hydrilla is an invasive species. And when we would go out and take these claws, in the middle of the lake, we started to see this stream invasive species, which is called corbicula, which is the Asiatic clam. It's known more for its stream occurrences than it is anywhere else. And I had a graduate student who figured out, who did the dispersion of this corbicula in Lake Seminole, and there's 5 billion corbicula, very conservatively, in Lake Seminole. Clearly, it's having some ecological role. In addition, we have a new invader, the apple snail, Pomacia, which eats hydrilla. So you have hydrilla, which is binding nutrients, anchoring sediments. You have corbicula, which is a filter feeder, which is probably also pulling out materials and storing it in biomass. But then you have the opposite trend of Pomacia, which is eating this. And we even have heard that some homeowners might be throwing Pomacia into the system so it'll eat the hydrilla and they can have a clean beach to swim in outside of their homes. This might be invasive species by homeowner. So very complex. So we decided, how can these things affect biogeochemistry? So we did some mesocosm experiments where we put corbicula, Pomacia, hydrilla, then combinations of the two. And this is the work of Stephen Shivers at UGA. And really, just to sum this up quickly, if you notice wherever we have hydrilla, corbicula, or hydrilla and corbicula, it has the ability to remove nitrate completely from the water column down to 0 in 5 weeks' time. Complete down to 0. So it has the ability to alter biogeochemical cycles. Again, think of how much hydrilla is in this lake. So clearly there's something going on, and whether or not the flows alter the hydrilla needs to be on the radar screen of what's getting through the system and towards the coast. So flows, hydrilla, nutrients. What about climate extremes, which are going to release a lot of water into this system? Killing off hydrilla, what's that mean for downstream Apalachicola? Reservoirs are capable of storing large amounts of these nutrients. It's not just land use to water to, or inland water to coast. It's land use to inland to processing to coast. And it's those processes and those connections that we gotta realize. So freshwater to coastal, take-home inferences, there's this idea of legacy nutrients. In shallow ecosystems, which is what we have in the southeast, we do not just deposit and forget nutrients. We resuspend those nutrients. We reprocess those nutrients. So the management practices of today are great at preventing new stuff getting in, but how the old stuff is being churned up needs to be understood, and how it's moving toward the coast. We need to understand these biogeochemical processes. I think the role of invasive species must be in this conversation. The hydrology seems to be driving all of this delivery. You need to know the hydrology of the reservoirs upstream from you. You need to at least have that in your back pocket. And then these cyanobacteria and toxins. When we think about the dead zone, when we think about what bacteria will do to dead cyanobacteria, they love it, and they can remove oxygen by consuming it. And these idea of legacy toxins, all these things need to be on our radar screen. They need to be in our conversations. And hopefully you've learned something about inland waters today, and we have some conversations moving forward. So I appreciate your time and your attention.
So I'm sure Matt would love to take some questions if you have them.
I'll start with the stupid one. What's considered a truly shallow lake system? Like, what's considered a shallow lake versus, something else?
That might be the number one conversation. Well, the number